The main goal of microarray image processing is to measure the intensity of spots or probes and quantify the gene expression values based on these intensities. This module will provide an overview of image analysis, which includes computer representation of images, addressing, segmentation, and intensity extraction, and then we'll look at the specific process used by Affymetrix. As a reminder, this is the general flow of a high-throughput genomic experiment. Once an array has been hybridized, there are various pre-processing steps including image analysis, normalization, and expression quantification. This week, we will discuss image analysis. Image analysis covers various domains including optical character recognition, or OCR, as well as identifying objects within a scene. This includes many military applications and surveillance methods for identifying objects in satellite photographs, such as seen in this example, where information from 3 sensor modalities yield data that are fused together to identify moving and stationary targets in a scene. In biology, image analysis includes magnetic resonance imaging, or MRI scans, outlining and measuring objects like stroke lesions in a brain image, as shown here, or when assessing structural deformations of the heart, assessing degeneration of retina, etc. We will focus on image analysis techniques as applicable for microarrays. The primary data from a microarray experiment are the set of images. Image analysis is required to translate the raw data, that is, the set of images, into numerical form. Recall that for custom spotted arrays, as seen on the right, 2 samples are independently labeled with different dyes and then combined and simultaneously hybridized to the same slide. This results in 2 images, one for the sample labeled red and another for the sample labeled green. Thus, the left-hand figure is a synthetic image obtained by overlapping the 2 resulting images. Alternatively, the Affymetrix gene chip is a grayscale image as only 1 sample is hybridized. The intensity of each spot is assumed to be proportional to the amount of messenger RNA in the sample. Therefore, images must be converted into quantitative data so that we can analyze the microarray data statistically. The steps for pre-processing and transforming an image into a format suitable for statistical analysis is called image analysis. Commonly, for a custom spotted array, the labels used are cyanide dyes, CY3 and CY5, which are distinguishable by the instrument scanning the array. Specifically, the emission wavelength of CY3 is between 510 and 550 nanometers, whereas the emission wavelength of CY5 is between 630 and 660 nanometers. Hence, we call these labels green and red, though they are not green and red in appearance to the naked eye. Therefore, the raw data for custom spotted arrays consists of 2 monochromatic images, one for the green dye and one for the red dye.